Hello, senior design students. My name is Massimiliano Pierbon. I'm an associate professor in this department, computer science and engineering, and I direct the molecular and biochemical telecommunication lab. Today, I will present to you a project that I'm proposing for the senior design cohort of this year. This project is entitled Webspace, a cybernetic platform for web information systems and space analysis. So first of all, I want to provoke you a little bit or tickle your curiosity with this concept that I don't know if you've ever heard. The Internet of Bio-Nano Things. What is that? So imagine a new generation of devices that is not only able to cover the human body and so sense and actuate possible processes inside the human body, but is also made of biology itself. Imagine engineered immune system cells. Imagine the possibility of having artificial organs, some of them even generated by your same cells. Imagine to have engineered gut microbes that work together with your digestive systems, and they might be introduced in your body through probiotics like yogurt. So these systems are today not science fiction anymore. They are becoming reality. They are studied in labs all over the world. Imagine also the possibility of having new tissue, maybe regenerated by your same tissue, that is repairing parts of your body or even substituting parts of your body with a better shape and better physical chemical characteristics. So all these systems are made out of what we call engineered cells. Now, if you haven't heard of engineered cells, that's your chance. I will explain you a little bit what they are. But imagine this system for a moment, seated inside your body and working together. Together with what? With systems that you might know or not know, but they have been already around for a while, like brain implant, cochlear implant, smart glasses. There's a lot of chatter about that now. Cardiac defibrillator and pacemaker, gastric stimulators, insulin pumps, and others like foot drop implant, like smart shoes. And all these devices are, yes, implanted or worn by you on your body. They work with your body, but they are electrical devices. So they work a little bit differently with respect to the cells of our body. Now, the main question is the following. Can we interconnect all these devices in such a way that they work together? They sense parameters together, they work together, and then, of course, they also communicate with other devices outside our body. What's better to spread your information regarding your health than the internet? I'm not talking about publishing on Facebook, of course, or having an Instagram post on your health, but imagine to be able to have a secure channel with a hospital or with your healthcare provider. So the very essence of this is this device that you see on the right hand side, is the possibility of making biology and electronics communicate together. So we call this biotic abiotic rebionic interface, and indeed here we have an electrode and here we have cells. So this is at the basis of what we call Internet of Bio-Nano Things, and this is at the basis of this grant. It's called the Rebionics grant, and with the Rebionics grant we are trying, and of course it's funded both by the National Science Foundation and by the Semiconductor Research Corporation, and my collaborators are at the University of Maryland College Park. We are trying to build this device that makes electronics and biology communicate seamlessly, but also by multiplicating this device into a board. Imagine like a circuit board. With one side there is a circuit, on the other side there is a biological substrate. Might be a tissue, might be a bunch of bacteria from our intestine. And so imagine to be able to realize in this circuit board communication between biology and electronics, two-way, memory, and computation. This is what we are studying now in my lab and in the University of Maryland College Park. Another provocation. So we talked about communication. Communication means the transmission of information between devices. This is a very, very broad definition of information. Information is a difference which makes a difference. Who said that? Gregory Bateson. He is one of the fathers of cybernetics. Imagine the internet. You send, you write an email, you send it through the internet, and then finally your recipient receives the email. The internet does not care what you write in the email, but just cares about delivering your email in the best way possible. So somehow these, that we can call them inorganic systems, in this system information propagates, but it's decoupled from what happens in the internet. If you write a love letter or you write a very bad email, internet does not care, does not change. So it's a little bit different for organic systems. In organic systems, information is stored in DNA genes. We see here a parsing of the DNA genes, functional parsing. Information flows through the DNA genes until becoming protein through transcription and translation. Protein is a molecule, but is also an informational structure because it's composed of these bricks that are amino acids that define, and the composition define the function of the protein. Information is also exchanged in the biological environment biochemically, okay, so between cells. So there is a slight difference here. Here information is inherently connected to the physics and the chemistry of the system. And so what we say is that here information is coupled to the physical and chemical structure of the living organism, in this case the cell. And so somehow there is a very, very tight bond between these two worlds that we don't see in inorganic system. And this spurred into the next grant I will talk to you about. But first of all, I want also to introduce you to the field of synthetic biology. What is synthetic biology? Is engineering cells or engineering biological systems. This is just a little cartoon that I took somewhere else, but this means that tomorrow means really, really soon, you will be able to type a code in a computer, like programming. You're able to hit print. Instead of printing on paper, you print on DNA, and then the DNA gets seeded into cells, and then the cells grow. So you might be able to have your own designer plants, for example, at home, or even your designer pets, which is also a little bit scary. This spurred into this other grant and this other project that is called Web Communication, WebCom, or Web Communication System, where we are trying to model the aspects of this communication where the system is intrinsically embedded into the semantics, so the meaning of the information. So if cells send emails one to each other, of course they don't send emails, but they send messages. Indeed, the content of the message that does influence also whoever transports the message. And so this project had many different aspects, also some STEM opportunities. So here is a short video of my lab that I want you to see, just to get an idea of what we do.
We're really interested in communication, and so right now we have communication on the macro scale and we also have it on the micro scale. We're trying to take that a step further and get it on the nano scale using biological systems.
In the Web Communication project, we want to study the peculiarities of these communication systems that are based on information that is transported by molecules and processed by biochemical reactions.
And the WebCode project is a software tool that's basically a framework to demonstrate how communication performs in biological chemical networks. So in this project, we try to actually create a framework for interfacing the biological world and the electronics world.
And we believe that our research will be fundamental to define a novel computing platform that is not based on electrical circuit and integrated logic, but it's based actually on biology.
We can use it for drug delivery or telemonitoring.
I am excited to explore the communication or information flow in biological cells.
So Dr. Pierbon is really helpful because anytime I have questions, I can go to him.
I continuously ask them, do you like the subject? Are you excited? Because I always tell them, okay, the moment you're not excited with what you are working, we have to change subject.
I like his energy and he's enthusiastic about research. So I'm happy to work with him actually.
Max is really good to work with, so he's like super fun, and also the enthusiasm here, as it's more like a childlike enthusiasm, so everything gets him super interested.
Biology is inherently complex, and the complexity of biology doesn't allow us to just have a model-based approach and everything works like electrical circuits. You have to be resilient, never give up.
Okay, so you got an overview of what we do. Some of the students that you saw in the video are actually coming from senior design. They were working on this senior design project, and so you see some of them here. And two of them actually started grad school right after the senior design, so they were very excited with what they did. So this is the first senior design team that I had. It was 18-19, and they also received an award, a gold award. It was awarded the Gold Award Team of the Year. And they developed this software is called wet code. This wet code was enabling chemical reaction network simulation in batch towards understanding informational aspects. So the second team that I had was just last year, and the project was called Paradox, where they built another simulator, this time to simulate the interface between electronics and biological molecules, which is called the redox interface. And so here you see them, and by the way, many of them worked in the summer. All summer, I actually hired them and worked with me to make this application even better. So let's go to the project that I propose. So you've heard about the Rebionics project, so communication between electronics and biology. You heard about the WetCom project, which is communication among biological systems in biological systems. So if you want to see the scope of the previous two projects that you saw, and evidently these projects have a lot of different elements that are in common. So the first, the wet code was focused on the biochemical network. So if you see inside the cell information, how it flows, and how it flows through chemical reactions, of course. The Paradox instead had a flow of information between the electrode that you see here, so electronics, and the biological molecules here. So what I have in mind for the Webspace is to actually wrap the two and make the two communicate. So the Webspace is somehow a closure of a suite of different simulation engines, and not only simulation but also estimation and design engines. But the Webspace will actually cover the aspect that is the most interesting, which is the estimation of the information content on the information flow and the design itself of the system. So for this, the Webspace project will actually take code base that has already been done and very well documented, so you have a very, very good base. You also have the possibility to talk to some of the students who developed that, because they are actually in my lab. They are grad students in my lab. And so take also the code base of the paradox and build a new code base that is based on two different main technologies. One is the System Biology Markup Language, or SBML. I don't know if you ever heard about that. It's standard, pretty well established. And then a little bit younger, this other technology that is called Synthetic Biology Open Language, SBOL. Libraries, software tools, there are many, especially for SBML, and some of them also SBOL. I know the developers of SBOL, so we will be able also to communicate to them. How is the architecture thought to be? Now, this is just a sketch for now. First of all, you will have an input that is an experimentally defined genetic circuit design and a redox-based biotic-abiotic element that will be given from the point of the Rebionics project. You will have a web graphical user interface front end like all the other software, and that will be fairly easy to make because you can use some of the code base that is there. And then the real part that transcends also research is estimating the information flow through the system. I say it's research, but indeed it's more of a guided development starting from the research that we are doing in my lab and our prior knowledge. So the Webspace will actually use application program interfaces to have a dialogue between these two projects, the Rebionics and the WetCom. This will be done through genetic part database, and tight communication with my collaborators, and then also biochemical simulation software. And we will also use other software, and this I mentioned, if you read the proposal, we will use the K-base of the Department of Energy to simulate metabolism. So this is the wet code architecture. Some examples of, this is the Webspace architecture. The name should be Webspace, not wet code. But indeed, it uses the wet code, is based on the wet code. Some examples are biosensing. Imagine to be able to control electronically the sensing of pollutants, but sensing that is done by cells, whose precision is unattainable with electrical sensing. Simulation of actuation of biological processes. Imagine to be able to control chronic diseases with electrical devices, but interface to the cells of our body. Augmentation of the functionality of the human body. Even more, being able to have a network inside the body that communicates information to and from a healthcare provider. Who are the stakeholders? Myself, and of course with my lab. William Bentley, Dr. Payne, and Dr. Godsey are faculty, are my collaborators, faculty in University of Maryland College Park. There will be a lot of exchange of information with them and participation to meetings, so formal meetings also with other faculty who have a different background with respect to mine, because they are actually biologists and biochemists and electrical engineers. Of course, the National Science Foundation of the United States and the semiconductor research corporations that are funding this research. Okay, so I hope that I gave you a good, at least a good first view of the project. What I can tell you is that the students who work with me were fairly happy. And indeed, not only they landed very good jobs after the project, but I continue to help them in their career, but also they built a CV that is fairly unique, because this research is done only here, only in my lab. And as you can see, there are a lot of point of contacts with the worldwide situation also, since it is a real exchange of knowledge and information between biology, healthcare, and computer science and engineering. With that, and then you will also have the opportunity to work with the Semiconductor Research Corporation, that is an interface between academic research and company research. So lots of opportunities there. You will be also having the opportunity to work with my grad students, so graduate students, and also with the graduate students and postdoc at the University of Maryland College Park. So if you have any question, don't hesitate to send me emails after watching this video. I'm just looking forward to this academic year. Thank you very much, and see you very soon.